Today we're Thank you, Lynn. So today's Journal Club we're going to discuss this recently published New England Journal of Medicine paper, SARS-CoV-2 neutralizing antibody um, LY-CoV555 in outpatients with COVID-19. So I assume that everybody has already read the article. So just briefly, the background of this trial is that no large randomized trials of SARS-CoV-2 targeted treatments was conducted intended to attenuate disease progression in early stage. And preclinical primate studies of neutralizing antibodies for SARS-CoV-2 showed marked reduction in viral load in upper and lower respiratory tract. So how do antibodies work against SARS-CoV-2? So we know that SARS-CoV-2 gains entry into cells through binding of its spike protein to receptors for the angiotensin converting enzyme 2 on target cells. Antibodies bind to the receptor binding domain of SARS-CoV-2, and therefore they can block the virus spike protein and neutralizing its ability to bind and infect. So we know that the antibodies were derived from coalescent plasma obtained from COVID-19 patients. And as you all know, that currently there's a coalescent plasma trial going on at Hopkins. And just briefly about the trial design. Patients were eligible when they're outpatients aged 18 years or older, recently diagnosed with mild or moderate COVID-19. And this trial is a phase 2 randomized placebo-controlled trial with 4 arms. Bamlanivimab at 3 different doses and with the primary and placebo control. The primary outcome is change from baseline viral load at day 11. And other outcomes included safety, symptom severity, hospitalization, and viral clearance. So, during today's Journal Club, we're going to walk through this article and discuss trial elements in the process. So here comes our first question for discussion about the eligibility criteria. So we know that all patients need to have a positive testing results for SARS-CoV-2 and presented with 1 or more mild or moderate symptoms. So could someone tell me how were mild or moderate symptoms defined? And also, why do trials need to clearly define the eligibility criteria? Okay, I'll start first. So I'll provide my thoughts. So this is how this is how they define moderate symptoms. So they adapted from an example from FDA. So FDA provided an example of the assessment of key COVID-19 related symptoms. So basically, they looked at different symptoms and they asked the patients what was the severity of your, for example, sore throat at its worst over the last 24 hours. And the patients would rate their symptoms as like no such symptom, mild, moderate, or severe. So my thought regarding why trials need to clearly define eligibility criteria is because of the target population. Feel free to jump in if you have other thoughts.
And also, what
Yeah.
might be some of the
Oh, go
benefits?
ahead.
Sorry, what might be some of the benefits for not having a clearly specific eligibility criteria in this type of study environment and this type of situation? Okay.
Yeah, so I was going to jump in with basically that. I mean, target population, you do want to have that clearly defined, but if you sort of go a little bit less clear or maybe strict on your eligibility, you can have a bit of a broader net, a bit more generalizable result potentially to people, and that might be more useful if you're trying to, in this case, you know, everybody is at risk pretty much. But you could go with a very targeted, you know, maybe people who have certain comorbidities that make them at higher risk, people who are at very specific sort of substages, in which case you might elicit sort of a better effect or some sort of effect just on that group of people, but you might not, you know, your target population then might not be as broad.
Yeah, indeed. That's very insightful. Thank you. Thank you, Riaz. Mm, okay. Also, another question for discussion is the placebo control. So do you think the placebo control is justified in the outpatients with COVID-19? How about in hospitalized patients with COVID-19?
I think it's going to depend on what you know about already for efficacy of other things, right? If this is right smack dab early on when people are throwing anything at the wall to see what sticks, maybe the placebo control, you know, that's okay. But if this is starting after people have a sense of maybe at least a couple things that might have some slight benefit or, you know, be able to help a little bit, then you should probably be giving that kind of whatever the current standard is as your control.
Does the allocation ratio have any influence on your opinion on that?
It's like 3 to 1, basically.
It is 3 to 1 in this trial, yeah.
Yeah, I don't know. It's a good question.
I don't know if other students have thoughts on that.
I think it's also relevant. Can you hear me?
Mm-hmm.
Yes.
I think it's also relevant in terms of how objective your outcome is. If you are using a really objective like outcome, it might be like less relevant for using a placebo. I think in this case, they're using a viral load. Yeah, so that may be less subject to any bias that might introduce in not having a placebo control. Yeah, and especially, but I think another point is that we're using the ambulatory like situations where like any patient encounter from this healthcare like professional might have a impact on the outcome. For example, if you are seeing the patients more, they probably feel better like already, like just being like able to have access to healthcare. Like,
so the question almost presupposes you know the answer, that in fact the trial treatment will work and will be relatively non-toxic. But if you don't know the answer and it turns out the trial treatment doesn't work and or is toxic, then the placebo control is absolutely justified. And even with viral load, unless you have absolute certainty about the natural history and time course of decline in viral load in an untreated group, then you need to understand, then you need an untreated control group. So I sort of think we tie ourselves up in knots about this. Where it becomes clear that you shouldn't is where there's a severe disease with a severe outcome and there's an effective treatment. That's just, you can't do that. But right now we have we have remdesivir of uncertain efficacy, but at least some perhaps. But we really don't have much in the way of effective treatment. And so I think we have to be careful to assume that we're assuming we know the answer in advance when we could be entirely wrong.
Yeah, thank you, Dr. Jepps. I think that's really important too. And I also agree with what you said and what Lynn said about the natural history. Like if we're absolutely clear about the viral load, like the natural history of the viral clearance, then maybe placebo control is justified. And I agree with Rias regarding the like how you choose the whether it's ethical to choose placebo controlled. It depends on whether you have available known efficacious treatment. And here's what we know. Thanks to Sharissa. So this is the NIH guideline regarding the treatment pharmacologic management of patients with COVID-19 based on disease severity. We can see that on the first row, if it's, sorry, on the first row is if it's not hospitalized patients, there's no specific antiviral or immunomodulatory therapy recommended. So I guess for this particular population, placebo is justified. And as Dr. Jepps mentioned, that remdesivir has proven to be efficacious for a certain type of patients hospitalized, require oxygen. So then for this group of patients, maybe it's at this stage it's no longer eligible, like no longer ethical to choose placebo as a control. The third question is that about the dose selection. So for those of you who read the article, so how was the doses of bamlanivimab evaluated in this phase 2 trial? How was it determined? And do you have any comments regarding how the doses were determined? So we know that 3 doses were tested. Okay,
I think
they say they use modeling, sort of pharmacologic modeling, to predict which doses. But I'm not sure sort of what kind of thing they're. I guess maybe they don't need to go further in depth into that. Sort of if you get sort of predictive the effect you might see, and then you sort of increase that. That's basically what you'd be doing. You sort of increase your dosage and see how much you'd be potentially impacting the outcome. And then it says they increase those by a factor of 10 to ensure adequate target coverage. So they're really pushing it. It's the kind of thing you do with like animal modeling. If you are trying to find like toxic doses, that you increase by certain factors to get sort of an efficacious dose, but not toxic dose. Generally, I think you actually decrease with animal, but yeah.
Mm-hmm.
And also, I think they have data from the phase 1 trials supporting that all of these 3 doses are within the maximum tolerant doses, even the 7,000 ones.
Yeah, thank you, Reyes and Lynn. So combining the answers from you, the 2 of you, I think we have a comprehensive picture here. So basically, the 700 milligram dose were based on, as Reyes said, the pharmacologic modeling. They estimate that this dose would be the maximum effective dose. And then they said, given the gravity of the pandemic, they increased the dose by a factor of 10 to ensure adequate target coverage and mitigate translational uncertainty in the model predicted PK and PD of the monoclonal antibody. And as Lynn mentioned, that it's also supported by safety data from phase 1 trial, which involved hospitalized patients. And as Reyes pointed out, usually doses tested in phase 1 trials were from like preclinical studies, from animal studies. So usually it's 10% lethality in mice. That's roughly equivalent to the human maximum tolerated dose, but they were justified by the body weight and the surface area of the body. I was wondering, do you have any comments regarding the increased by a factor of 10? Is that common, or it's like it's kind of desperate under the pandemic? Like desperate time calls for desperate
Yeah, I'm not
That's-
really sure, because if 700 is the maximum effective dose, I'm not sure what increasing that by 10 is supposed to, what the purpose is supposed to do, I guess.
There's no scientific rationale behind it, quite frankly, but it is standard procedure. That's typically how the dose finding is done, unfortunately.
Well, remember the maximum effective dose is based on models, and so
Mhm.
those models are frequently wrong.
Yep.
So especially in this case, they want to make sure that they don't exclude the possibility of a dose that would have been effective if 700 turns out to be too small. But I'm struck, there's a comment in the article that says there were other treatment groups, but they only reported on the 3 doses plus placebo. I don't know what those other groups were. Is that coming up,
Yeah.
Lynn?
They didn't say that in the article.
Or say
Let
what
me
they
check
were.
the-
Yeah.
Although the trial contains additional treatment groups, here we focus on the interim analysis of results from only 4 of these groups.
Mhm.
So I just checked the protocol. The protocol didn't say which other doses they tested. So like the protocol was published as appendix to the New England Journal of Medicine paper. So maybe
Mhm.
they have another protocol, but like I'm not sure which doses they tested additionally.
Okay. Well, I would think as just as good practice to have, they would be in the protocol. So
I doubt the other doses are greater than 7,000. They're probably in between somewhere and had small sample sizes. That's what I thought when I read this.
Yeah, that's a really good point. So, okay. My next question is that regarding the treatment regimen, could someone tell me what was the timing of the treatment administering? And do you have any considerations regarding the timing?
I think that was within 3 days after the positive test. I do have some concerns about the timing for the primary outcome because it says 11 days after the positive test. So it's not 11 days after like randomization or transfusion. And there is a window which is plus or minus 3 days for the 11-day outcome.
Yeah, indeed. I have the same concerns. And when I was discussing it with Teresa, and then we looked up and there wasn't a clear defined randomization time point. All we know is that the treatment administering needs to happen within 3 days after the positive results of the testing. And here I'm showing the design outline from their protocol. So there's that the patient who have a symptom would get test swab taken. And then when the results came back, they would ask the patients if they were willing to participate in the trial. And if yes, then they randomize the patient at some point and then administer and give patients the infusion. So that is all we have. And regarding the consideration, I was wondering, So I was thinking of the start of symptoms. I mean, the patient can get tested at the early stage of this, like at the first day of their symptom, or at 3 days or 5 days after the symptoms started. So do we have concerns regarding like patients are actually at different disease courses, like different time point?
Severity. Mhm.
And the the trial did try to address that. So what they did is that they stratify by the duration since the symptom onset. Yeah, I guess it's a like, at first I was thinking of, can we do like the timing of treatment administering, can we set it as within some days after the onset of symptoms? I don't know what you guys think of that.
You mean restricting randomization to those within a certain amount of time?
Yeah.
Yeah. Can I chime in?
Sure.
Yeah, in terms of maybe the internal validity, I mean, the comparability of the 4 groups, I don't think as long as the randomization is like, we had a rigid process, a robust process with the randomization, I think the 4 groups will probably be comparable in terms of how like the distribution of the natural course of the diseases will be like comparable among those 4 groups. But it will impact our like how the conclusion of the trial might be applicable to the general population suffering from the disease. But unless we're in a very controlled setting, maybe a challenge trial where we know exactly where the exposure date would be, this is as bad as it gets. Like most people will go to the hospital according to maybe the anchor date would be the day they started having symptoms. So I think maybe the trial is doing a decent job in terms of that.
Yeah, you brought up a really great point. Actually, when I was thinking of which journal, like which article for this journal club, I before this trial result came out, I was thinking of the challenge trial, that like the ethical part, but Riaz has already talked about the ethics of challenging trial. I don't know if any of you has any comments regarding that, although it's not the, yeah. Okay. So we talked about the treatment regimen. So the follow-up scheme of the trial is that they follow the, so they have this study day and different visit type because it's outpatient. So they have on-site visits, and also they have the telephone visits. Here are the set of their outcome, the full set of their outcome. So we have primary outcome change from baseline viral load at day 11, and some key secondary outcomes: safety, symptom severity, clinical outcomes, hospitalization, and deaths. So what do you think of the outcome set? And I guess the follow-up question is that how would the outcome set, measurement set, differ if we're interested in like hospitalized patients?
Well, so this is a phase 2 trial. And the use of viral load to say we think that this is going to work is a reasonable primary outcome. And it's also one that enables you to have more events so that you can do it with a smaller sample size. If you move to a phase 3, you're going to want a clinical event, which would probably be hospitalization. That's a much larger trial, with viral load as perhaps a secondary outcome. Once you move to hospitalization, you have to use a different outcome, and that's probably might be duration of hospitalization, admission to an ICU, intubation. I mean, there are all sorts of different outcomes that you can use. The simplest one is duration of hospitalization, because you have more events, meaning you can have a smaller sample size. But for phase 2, a biologic marker is fine. For phase 3, you're going to want a clinical outcome, my opinion.
Yeah, I totally agree.
May I ask a question here about this? This is Jay. I was confused. I agree with the value of a viral load, but they're using, we can get to this later, repeated measures analysis. And that implies there are more measures of viral load than just baseline and day 11. Because otherwise, because if there were only those two and you're taking the difference, you wouldn't use repeated measures analysis. So does anybody know if they measured viral load in between the baseline and day 11?
Yeah, they did. So they measured the viral load. So in the results section, we will have a slide. They also measured at day 3, day 7. So yeah, they actually have repeated measures. That's why they use the mixed effect.
Okay. Yeah, thank you. Thank you. Good.
And also, okay. So here is and thanks to Sharissa, here is a set of outcomes that that is recommended by the WHO. So they proposed a core outcome set, which include viral burden, survival, and clinical progression. And as Dr. Jab said, so if we are interested in, like, if we're conducting a phase 3 trial, or if we are focused on hospitalized patients, then you could have all these different outcomes that you could potentially look at beyond viral load. And here the duration of hospitalization was not mentioned. I guess this is not complete, not comprehensive enough. Okay, so regarding disease severity, why do we need to clearly define it? So from a, let's say from a regulator point of view, so if you're FDA, why do you want like clearly defined disease severity as an outcome? Like for phase 2, for phase 3, and you have multiple products potentially effective that then you can approve. So what's the value of having sort of standardized, clearly defined disease severity?
As an outcome?
Yeah, as an outcome.
You go ahead, Riaz.
No, you go ahead.
Oh, it's probably easier for like whoever's doing that aggregating data in FDA to actually have something that is codable. Like, yeah, it's easier to compare across different trials and use aggregate data.
Yeah. Go ahead, Riaz. You're going to say.
Yeah, I'd say sort of for defining it, I could see maybe there being some utility as well if you've got it clearly defined as an outcome, helping to differentiate what the end result is for the drugs, for the intervention, see what the effect, if you're not really being clear on your definitions of disease severity and somebody comes out of it, maybe 2 people come out of it, it could be difficult to differentiate which one of them ended up sort of in the better pool. But if you've clearly defined all the different stages, then you have a bit of a better sense of, sort of some sort of categorical ordinal set of have they actually improved or not.
Yeah, indeed. So the 2 points that you, Riaz and Ling, you brought up complement each other. So like as Riaz said, there could be different patients, like the heterogeneous nature of the symptoms. in outpatients. So to Lynn's point, like we need to provide an accurate evaluation of the benefit, and in the end of the day, we need to decide whether to approve A drug or B drug. That's why FDA provide this guidance for assessing the symptom, like COVID-19 related symptoms in outpatients. So that was the table that I showed before.
Lynn, are you going to talk about the EUA now or a little bit later?
A little bit later.
I'll save my comment then.
Sure. So about the question, here's a question for sample size. So how was sample size determined? And what are the considerations when determining the sample size of a trial? And this is the information provided by the authors.
So I actually looked this up, and they used this dynamic viral model and simulation to calculate the sample size. And I think one of the reasons for that is the primary endpoint is varying by time. So by using simulation, it will be better to calculate a more accurate sample size as compared to the conventional sample size calculation method.
Yeah, that's a very good point. Thank you, Shang-Ji. I don't know if Jay has any comment regarding this.
Yeah, Jay wrote the whole statistical analysis plan. So.
Yeah, I don't have a comment. I think we should remember it's 100 patients per arm, but then they also combine the doses at different points to any dose versus placebo. So then it's a 3-to-1 comparison. So they're getting at least this much power when they test just a single dose against a placebo.
Yeah, that's a very good point. Yeah, they pulled the doses so they will have even larger power if what they assumed the detectable difference is true. So yeah, and when we are calculating sample size, like we need to consider like the detectable difference, the power, the alpha level, depletion, like loss to follow-up, and also as Shang-Ji said, if we have if our algorithm is changing over time, it's better to use a dynamic model like simulation. That's require statistical expertise to do that.
Can I add one thing? Just a reminder that sample size is based on multiple guesses, and frequently those guesses are very wild guesses, even unfortunately in phase 3 trials, and I think certainly quite often in phase 2 trials. So just to remember, those sample sizes, they sound very precise and technical, but the underlying numbers are often just guesses.
Yeah, that's a very good point, and we will come back to your point. So, and the further questions so when we look at the results, so here's the first figure that they gave us. So we have about 100 patients in each treatment group, and about 143 patients enrolled in placebo group. So in terms of result reporting, is this something that you will typically see in a trial report? And is there anything missing?
So I suppose it's a one-time treatment that makes it so you don't have to worry about the adherence and dropouts and something like that.
So yeah, let me ask you, What do we typically use to report patient flow
You mean in an interim analysis?
in trials?
I think the first will probably be the I mean, after like the comparison of the different groups, the table one, I think next would be the how many patients actually stick with the original assignment and how lost to follow-up, something like that. The performance of the trial.
Yeah, indeed. And what's that graph? Counselor diagram.
Yeah. Okay.
Yeah, this is the really good talking points brought up by Sharissa. So this is not typically what we see in a clinical trial. And yeah, I agree with you, Lee. So maybe it's because of it's a phase 2 trial. But still, like, I think they can do a better job, like reporting the randomization, which people are included in analysis. And although there might not be adherence issue, there may be like follow-up, like deviation from follow-up, like patient miss visits, kind of.
Yeah, because how did they define their analysis set? Do you guys remember? Was it
Yeah,
true?
they require one, at least one follow-up visit after the treatment.
And they also required actually receiving the treatment. So those are two criterion added to the randomization.
Yeah. My slide. So here are the table one. So just want to so the patient characteristics were well balanced between the treatment and control group. And they mentioned the risk factors for severe COVID-19. So basically 70% of the patients would have a risk factor for severe COVID-19, defined as an age of 65 years or older, BMI of 35 or more, or at least one coexisting illness in certain specified categories. But however, I failed to find, like, even in their protocol, what those coexisting illnesses are. And in terms of disease status, so we can see that 75, 79, like 80% of the patients had mild symptoms. And the median number of days since the onset of symptoms were four days. So these patients like got tested really fast, and the test results came back really fast. Okay. So the primary and secondary viral outcomes are here. So we can see the primary outcome is the viral load, change of viral load at day 11. And the results show that only this 2,800 milligrams group have a significantly lower viral load. Like the difference in change is significantly different when they compare to the placebo group. And also, when we look at the mean change in viral load at day 3, again, this 2,800 milligram group was significant. However, at day 7, it's not. And for other dose groups, the results are non-significant. For pooled doses, we have one significance here. So I don't know if you have any thoughts or comments here.
Well, each day is plus or minus 4 days, is that right? Not just day 11, but the intervening days also are plus or minus 4 days, depending on when they could get them in. So they lose a lot of precision here. I mean, it seems to be suggest an inverted bathtub response, with the middle one being the significant, but on the other hand, it could be a function of they're not getting getting people at such what I would consider a wide range of days between. I mean, plus or minus 4 wouldn't matter that much if there were 60-day follow-up, but with only 11 days, there's overlap. And it's also last observation carried forward. I don't know what they mean by that. They might mean that even something outside of the plus or minus 4 at the 11 days, something earlier visit is automatically made day 11 because the patient doesn't have a day 11. That would be pretty sloppy. I think the whole description of that we are looking at the change from baseline to day 11 is a sloppy way of describing they were doing repeated measures. So it's really the slope. I mean, ideally, they're measuring the slope between day 0 and day 11 and how that differs between the treatment groups, between these doses. I don't understand why they word it the way they do. It looks like only 2 observations are involved. But I think there's a certain sloppiness of because of the plus or minus 4 that may be responsible for some of these doses not being significant.
So
Yeah.
I look at these data and I take away 2 almost contradictory messages. The first is there's no dose response curve. The 7,000 milligram dose is actually no better than the 2,800 milligram dose. And that's worrisome. One would at least like to see something, either a plateau or some sort of dose response curve, you know. The second is, though, that all of these numbers, if you look like, for example, at the viral load, well, even the 7,000 is no better than placebo. So unless they have a spectacularly good reason why 7,000 should not work, whereas 2,800 were, I'm a little worried we're seeing a lot of noise and not a lot of signal.
Yeah, that's what I'm saying too, but in a statistical way.
Well,
Yeah.
Go ahead.
Go ahead. Yeah, I just want to say that really cleared the picture for me, because I was wondering why we are seeing what we're seeing.
I have a question and a comment. My question is, I'm no expert in viral load. Are these differences in viral loads impressive, likely at all to be clinically relevant or not? I mean,
that was my question, especially in the context of COVID-19. I don't know how relevant that outcome really is. Not that it's relevant, but. Yeah.
Right. We talked earlier about using this as a relevant outcome for a phase 2 trial, but it still would be nice to be able to put it into some perspective. I don't know if a difference between minus 4.0 and minus 3.47 is meaningful at all. And then the comment is just, when you do these multi-dose trials, and Sharisa and I have worked on one that's very similar, it's almost like a U-shaped response curve here if we trust these point estimates. And so that makes it even harder to figure out what to do, but certainly would argue against paying any attention to pooled doses, it seems to me.
Yeah, another good point too. So yeah, I don't think I have enough expertise to answer that question. So just what the article said, so the whole population, the log viral load for the entire population decreased by 3.81. This value corresponded to a decrease by more than a factor of 4,300 in the viral burden, for elimination of more than 99.97% of the viral RNA. And the difference between like the 2,800 milligram to the placebo, so the minus 0.53, is corresponding to a lower viral load by a factor of 3.4. So I guess the 3.4 versus the 4,300 fold, it's like 1 out of thousands difference. So maybe, I don't know if it's clinically, significant.
And that drop in viral load is also consistent with the disease trajectory in general. So it's really hard to make sense of this.
Well, so, just thinking about viral load drops. With HIV, you drop, well, first thing is what's the lower limit of the assay detection? We, in the HIV epidemic, we had improving assays over time, but originally 400 was was considered undetectable because of the quality of the assay. So that's the first question you have to ask is sort of, what are the limitations on the assay? Second, we would see drops from 10 to the 5th down to 10 to the 1st, so 4 log units, and that would be a very meaningful drop. The problem you have is that with placebo, you're seeing drops that are pretty substantial, and the marginal benefit is pretty modest. Looking here. So you combine with, at least in my case, uncertainty of the quality of the assay and its detection, and a pretty modest benefit, and a U-shaped curve. Every drug I've ever seen that has a U-shaped phase 2 curve, and the company goes after the middle dose, turns out to be either not effective or not very effective. So again, like I said before, I'm very worried that there's a lot of noise here and not a lot of signal.
Yeah, that's a very good summary of this question. Moving on to other results. So the article also showed some exploratory measure of viral clearance. So basically they showed the hospitalized patients represented by the red dots have generally higher viral load and slower viral clearance compared to the non-hospitalized patients. And from the second graph is showing different doses versus placebo regarding the viral load. So you can see the 2,800 milligram, the blue curve, has like higher probability of having low viral load patients. And then speaking of the hospitalization, so here are the results. So the hospitalization rate in the placebo group was 6.3%, and in the pooled treatment group is 1.6%, and it's significant. However, here, as we have talked about, the benefit in hospitalization was not shown in 2,800 milligram, because perhaps it's like the hospitalization rate is really low and it's kind of a rare event. Maybe they will see something if they have phase 3 larger trials and a better dosage. And then in the post hoc analysis examining hospitalization among patients who were 65 years of age older and among those with a BMI of 35 or more, the percentage who were hospitalized were 4% in the treatment group and then 15% in the placebo group. So the treatment effect is more significant in these high-risk population. And then here are the symptom scores. So remember, we have assessed patient symptoms daily. So they show that from day 2 to day 7, the treatment group the pool treatment group has a lower symptom score. So basically the symptom is better than the placebo group. However, from day 7 to day 11, it's not that obvious because the patients in both groups basically fully recovered with only very mild symptoms. And then here are the symptom score by those different dosage, and not surprisingly, the pattern we see in the viral load is not consistent, is not seen here. So the light bluish green line for the 2,800 milligram is actually higher than the 7,000 milligram. So in essence, the viral load and the symptom score, they're kind of telling different stories. So I don't know if you have any comments here.
Yeah, I mean, I think this is sort of a good example of one of those things where, I mean, so the classic, do people just get better on their own as well over time? It's sort of showing, there is a bit of a difference there, right, between sort of the treatment and the placebo groups, but the trend is certainly placebo people are decreasing quite fast still. Their symptom change from baseline is still quite large, as the days progress. And so is the difference between placebo and intervention really that significant, with the added risk potentially of other adverse effects and things for how the people are doing?
Yeah, that's a very good point. So that speaks to the value of this treatment among these patient groups. But I will ask you the question of approve it or not later.
Well, okay.
I guess. Go ahead.
No, you
Actually,
said you're going to ask a question later, so I'm not going to talk about it now.
Oh, okay.
So let's think from the perspective of the drug company. I mean, after phase 2, they got to make a decision, go, no go for a phase 3 trial, right? And they also have to make a decision about, if they're going to go, what is going to be the indication, i.e. in which people are they going to try it? And they have to make a decision about what's going to be the clinically meaningful outcome, although the FDA has provided some guidance on what might be. But think about it, how big a trial it's going to have to be if they go for hospitalization. Yes, there's a 2/3 reduction in hospitalization from 6 to 2%. And that might be meaningful in a high-risk group, but there are all sorts of decisions about drug development that enter in at this juncture, not just approval, but drug development and what are you going to do with these data?
Yeah, from both the regulator and the company's point of view, yeah, that's really good, insightful.
And it's not really clear to me, at least, what the benefits of Operation Warp Speed are from an industry's perspective.
Mm-hmm. That too. So, okay. So for the primary outcome, yeah, we sort of talked about it. So, yeah, we went through it. It's the change of viral load at day 11, but plus minus 4 days after positive test measured by a nasopharyngeal swab, followed by RT-PCR. And only the 2,800 milligram dose group showed significantly lower viral load at day 11 compared to placebo. And all dose groups showed treatment effects on symptoms at the earlier time point. So so what did the authors discuss about the viral load as a primary outcome? So they said these may not be clinically meaningful because viral load was substantially reduced for most patients, including those in placebo group. That speaks to the clinical significant difference that Dr. Dave Shade mentioned. And they talked about RT-PCR may not be the accurate measure of viral neutralization because viral RNA may persist, but the replication competent virus may not be there. They also talked about nasopharyngeal secretion may not be the ideal specimen because the severity of illness was driven by lung injury and lower respiratory tract. And here's a question about sample size. So among these, like, so do we think, so they talked about, let me see. Oh, here. So was the study sufficiently powered for significant result? So they assumed, like as Dr. Dave Shade mentioned, that it's wild guess. So it turned out that in placebo group, their guess is pretty good. But in the treatment group, it seems that they overestimate the treatment effect. if you compare these to the 4.38. So like they don't really have enough power to detect significance in other dose groups. And also the safety results are insignificant. So basically the serious adverse events, so we're not observing treatment groups, and any adverse events, the proportion of any adverse events were comparable between placebo and treatment arms, and the most common adverse event was nausea in treatment group, so slightly higher than in placebo group, so it's not significant.
Can you, Lynn, can you go back just one slide? I just want to make an observation here. Zero serious adverse events, yet they report hospitalizations. And by most standards, a hospitalization is always a serious adverse event. So they've either defined serious adverse event specially, or they're reporting them over a different period of time that excluded the window for hospitalizations. And that can be, I think it's okay to exclude hospitalizations for your primary outcome in a table like this, perhaps, but it ought to be explained carefully.
Well,
Yeah.
Dave, but remember, hospitalization is an outcome. So
Right.
You can't, but, and it's presumed to be an outcome of the disease, not an adverse effect. So I'm not so sure that I think you got a problem with calling it an adverse effect in a study like this, where it's an outcome due to the disease.
Yeah, I don't necessarily disagree, but here they're calling it an event, not an effect. And from a regulatory standpoint, there still are the reporting requirements unless you've got a clearly defined policy that excludes outcomes or events that you have pre-specified. And even then, you know, it's a guess as to why someone is hospitalized. So I think I would tread carefully in that regard. Excluding hospitalizations, I think it's possible, but it's risky. And I think it should be just clearly explained, that's all.
Yeah, I think I agree with both of you. So the conclusion, so they came to the conclusion, so I think they have a rational conclusion. So they said at least patients received the treatment had fewer hospitalization, lower symptom burden than those who received placebo, with most pronounced effect observed in high-risk cohorts. And if these results are confirmed in additional analysis of this trial, this drug could become useful treatment for emergency use in patients with recently diagnosed COVID-19. So here comes my question. If you're FDA, will you approve this drug? Will you give it an emergency use authorization? What's your rationale?
Can I change the perspective? Like,
Sure.
if I'm the drug company, I'll probably look at the data with subpopulation that has comorbidities. For example, if they have like asthma patients in those 400 patients, and somehow like in the intervention group, we're seeing much better results, I'd probably focus on that and stop here with the general like enrollment and just focus on asthma patients. But I suppose like the safety profile probably differ, you know, the asthma patients having more propensity to like allergic reactions to the antibodies, but it's probably a better angle, like considering what we have so far.
Can I ask a follow-up question?
Sure.
So does that, so you, if you were the drug company, you would like to limit your potential audience, your customer base, to a smaller group so that you could sell it to less people?
Yeah, because I think based on the interim results, it probably won't be approved for further trials in phase 3. Yeah, but yeah, just get as much as we can, protect the asthma children at least.
Will you try to apply for like a full population first, and if you fail, then you narrow the population?
Yep.
Okay.
So, you know, remember, there's a cost to going for the widest audience possible. And the cost is the cost of doing that trial. And if they think that it's a huge trial with a marginal benefit that has iffy approval process because the benefit is so marginal, they might go for a high risk group where many more events would mean a smaller trial. If the efficacy is similar, a reduction in hospitalization by 2/3, and that leads to approval, generation of a revenue stream, and the opportunity to expand its markets later on. So these are all development strategies that you have to think about. Approval sometimes is actually, it's never easy, but it might be easier. Would I approve based on these data? I would, if I were on the council, I would vote no. But on the committee, I'd vote no. But you know, that's, nobody is going to go for, you would not traditionally go for approval based on phase 2 data like this. You would like something that's a little more clinically meaningful in a phase 3 trial.
I saw
How about
and I saw Doug say no. What kind of approvals are you guys talking about? Are you both talking about general approval or emergency use? And does that change your answers?
I think we're talking about emergency use
Mm-hmm.
for a high-risk group based on just these data. They'd have to do a phase 3 trial with a single dose that they would choose, phase 3 trial with all of the risks involved to get a more general kind of or a regular approval.
Yeah. Yeah, but Jay, we would need to see those data, right? So I think based on this, just going for a high risk group, that will probably be pretty dicey. And I think, you know, my personal litmus test, would I take it with mild disease on an outpatient basis? I don't think I would. I wouldn't take it based on these data.
You think you would not take it?
I would not take it. If I had mild disease for an outpatient, I would not take it. Anybody here would?
In the absence of availability of convalescent plasma, I might. You know, so a 2/3 reduction in the risk of hospitalization, 2/3 reduction of a low risk, but I might.
Yeah.
Because I see no safety profile that makes me worry that there's a downside.
You would have to consider the alternatives.
Right,
Yeah,
right.
you have to. I mean, I think with convalescent plasma, we see, you know, a pretty good effect, and so there are options.
Right.
That's why I
And
wouldn't
it's
go
free.
for it. Lynn?
Lynn.
yeah, and
Lynn Nan. Lynn, is this the drug that Chris Christie got in expanded use?
I don't think so. I think he got the convalescent ones.
No, I think he got an antibody. He got an antibody, but I thought it was a different one. But I'm not sure, Janet.
It began with B, but you know, after that.
But I thought he got a monoclonal antibody and not convalescent.
Yeah, because in the article that Lynn had sent me, he got it after refusing to be in a clinical trial. And then the clinical trial showed it wasn't useful for hospitalized patients.
Okay. Thank you all for your comments. So here's what FDA did. They gave the drug emergency use authorization for to treat mild to moderate COVID-19 in adults and pediatric patients aged 12 years or older with positive viral testing and who are at high risk for progressing to severe COVID-19 and or hospitalization. A slightly broader population in terms of age, but narrower in terms of risk, so high risk population. And currently this treatment is for free, so no out-of-pocket cost for patients. Have any final comments, thoughts?
No,
Very good discussion.
yeah.
Thank you. Thanks everyone
Thank
for
you.
coming.
Thank you everyone.
Right.
Thank you, Teresa.
No problem. Great job, Lynn.
Thanks, everybody.
All right.
Good job, Lynn. All right,
Bye.
take care, everyone.
Chris Christie did get this drug. I just found it.
Great.
And he thought it was cool. So.
Yeah.
Well,